Anupana occupies a unique place in both classical formulations and modern integrative therapeutics. I now invite Dr. Satish Pai to deliver the scientific session. Over to you, sir.
Pranamas to everybody. Am I audible?
Yes, sir.
Okay.
Loud and clear.
Yes, sir.
Yeah. Let me begin my presentation. It's a long way since 2020. Honorable Jayashri Madam asking me for presentation on honey. It's 2026. I'm glad to inform that 6 more researchers have been included in line to this my long journey in the research field of Madhu. Let me begin with my presentation on the topic therapeutic potential of honey. It's a research evidence and emerging clinical applications. Before going through, my humble pranamas to my aptas, my gurus, and Dr. Shailesh Nadkarni, unfortunately, is no more. He is the person who inspired me in the field of research, but I still remember his word that you must also concentrate on clinical applications as well as clinical studies, clinical trials, uh, because basically I was more interested in analytical and experimental works, and Sir's inspiration, Sir's words inspired me to take up clinical studies also. So with gratitude to my gurus, Dr. Jayashri Madam, as well as Dr. Shailesh Nadkarni ji, whose guidance became the foundation of my journey and whose blessings remain my strength. Now, before moving to the subject proper, some historical aspect previously explained is the Madhu is one of the earliest known superfoods mankind knows, and the literature goes back to 1200 BC. We still get some references, even the Ayurveda, which is much more ancient than that, and the Rig Veda, around 300 times, the concept of Madhu has been elaborated. This is one, the ancient or much old sample of Madhu excavated, and honey never spoils. This sample of honey is found 3000 years back, the old honey in the Egyptian tomb that was still edible. So Madhu never spoils. And basically Madhu is also called as Pushpasava, a substance produced by the Makshika or the bees. Madhu is basically Medohara Dravya. It is known for its Yogavahi karma, we will see how this Medohara karma, Yogavahi karma have been evaluated, have been found and proved in the what we call the translational research. Okay. Madhu is basically produced by the enzymatic reaction of bees saliva. So if we consider this is just a bees omitus, but later on, which becomes a nectar for mankind. Some nutritional informations. The honey contains around 200 substances, includes sugar, water, organic acids, vitamins, minerals, pigments, phenolic compounds. I keep on mentioning this word again and again because this will come till my end of my presentation. The volatile compounds are also been found in Madhu, and then sugar and water. Basically, sugar amounts up to 95.99% in dry honey matter, and there will be D-fructose, D-glucose, which represents around 85 to 95 of the total sugars. And some of very important components of the madhu includes fructose, glucose. See, all these things I am telling you regarding, because this information is going to be, useful in some of the clinical applications also. And very important uh, ingredient of madhu is something called as HMF content. This one, the HMF content, this is nothing but hydroxy methyl perfurol. Why this HMF content is useful and how it is going to be, you know, regarded as a quality component to assess the quality of the mud. Now, there are few types of honey are also being described in our classics, but the only honey what are available today are the one is Makshika Madhu and the second one is Brahmara Madhu, what I have seen. The Madhu is the one which I have seen, since my childhood. So just by odor, can recognize whether it is adulterated, whether it is genuine, because of the experience of having or rearing madhu in our house. So Makshika Madhu is said to be the Madhu Shreshtham, the Shrestha Madhu, and one of the key feature of Makshika Madhu, which is described as it is Tila Tailavat. So that is the feature which makes the identification of Makshika Madhu. Otherwise, the second sample available, other varieties of madhus are not very much described, very much distinctively seen. But Makshika and Brahmara Madhu is very crystal clear. As in the classics, it is been told like it is Sphatikavat. So that is nothing but the Tudve, or even one or other variety which is available is called as Misri, or a very small bees, stingless bees. They are also called as stingless bees. The honey of that will be very crystal clear, without any color, without any pigments. But however, that nature of the honey is extremely sweet. Now, some of the gunakarmas, because these are the gunakarmas which pave the clue for us in conducting the researches. So, one of the gunakarma what I considered for my research all through this year is Vilepanam, Saukumaryakaram, Sroto Vishodhanam, Chedi, Vranashodhana Ropanam, Vishadam, Rochana, Prasada Jananam, and very important, which is called as Yogavahi. This Yogavahi karma is often been regarded as a synergistic activity, but however, Yogavahi is something else apart from synergistic activity, which we will see in the future slides. There are some differences between Navina and Purana Madhu as well described in our classics, because this concept was also considered for the research. For all kinds of researches, what we did in our institution with the help of postgraduate scholars, the Purana Madhu is the one which was more or less used. Purana Madhu is nothing but the Madhu procured and which is aged for at least 1 year can be called as Purana Madhu. And one of the key feature of this Purana Madhu is Adilekhanam. This Purana Madhu possesses, as the age advances, we still have the honey sample which dates back to 2012 2013. We have kept it. As the days advance, the honey samples become sugary, or it precipitates, it becomes crystalline. But still, there will be some differences with its gunakarma, with its odor, with its color, etc. Likewise, the karma is also going to be changed, but however, the quality of the madhu remains intact. So one thing to remember always, the Puranam Ati Lekhanam. So if we want to achieve the Lekhana karma from madhu, the best way to use is Purana Madhu. Procure it, store it for a period of 1 year, then we can use it for the purpose of treating or using for Medohara karma. Now, the very interesting thing what I got from the book Kayadeva Nighantu, because this information is not seen otherwise. Uh, we all know that the heating of madhu is contraindicated. See, you ask any person from Ayurveda, they will definitely say the madhu or honey should not be heated, and heating will cause its uh, it becomes poisonous, right? So now why should not heat it? Because there is some contradictory remarks have been given in Hayadeva Nighantu in the first 3-4 pages of Kaideva Nigantu, we will get the reference. For example, if we go through the shloka, that is Chaudasya Pake Cha Ashtamshe Namamamsham Jalam Kshipet Durmamse Mutra Samyuktam Sthavletu Trifala Rasam Ropane Kshira Samyuktam Shodhane Rajani Rasam. That means to get some desired clinical effect or desired clinical efficacy with the help of madhu, we need to subject madhu to madhupaka. That means heating is indicated whenever there is a therapeutic applications are concerned. Otherwise, if the madhu has to be consumed or administered in the form of food, the heating is not indicated. So this is some differences within our classics itself. There are references madhu should not be heated, and there are references that without heating madhu should not be used for the treatment. Go through the shloka again. That is sarvarogeshu paniyam datva mradvagnina pache. If we want to use madhu for therapeutic purpose in all rogas or all vyadhis, so sarvarogeshu paniyam datva Madhvagninam pachet. So the heated version of madhu is indicated therapeutically. Kshiptam jale nimagnam syad athava pichilam bhavet vastre darvya vilepeti madhupakasya lakshanam. So till when or till what point, what is the end point of madhupaka? Madhupaka is nothing but until it turns into avalehavat. That is shipte jale nimagnam. When we immerse or drop a drop of that pakwa madhu in water, it should just sink, or it should get when we apply it over the cloth, it should stick on the cloth. Then the darvi or the mixing the vessel, it should stick to it. So vastre darvya vilepeti madhumakasya lakshanam. So madhupaka is the one which ignited or which draw my attention. So why can't we take this particular concept of madhupaka for the upcoming research, among which the ropana karma, then Ranashodhana karma, then Durmamsa, that is anti-tumor activity of Madhu, as well as Sthaulya, that is anti-obesity, as well as anti-hyperlipidemic activity. All these uh, areas we have tried, and some results I want to present in front of you, which can be further applicable for the clinical practice. So some pharmacological activities of the madhu should also be known before we proceed further. The Madhu is said to be very good antioxidant. Madhu has a very low glycemic index, so it is appropriate sugary substance or appropriate substance with carbohydrate to be used even in case of diabetes. And the honey reduces cardiovascular risk factors. And one more type is most of the honey have been proven anti-inflammatory also. And prebiotic, some more aspects of the madhu should be known. That is, it is prebiotic activities, uh, which is now being worked with one of my postgraduate scholar. Madhu has a very good synergistic activity: anti-metastatic, anti-tumor, anti-cancer, anti-proliferative, antibacterial. All these activities we have, we have conducted the works. And the one of the key factor which is attributed to the madhu, that is because of its low pH, which reduces the growth of bacteria and increases the tissue oxygenation. See, this is very important. Madhu is usually acidic in nature. It should be, because purana madhu, there will be some fermentation process happens, and there will be some generation of even hydroxide hydrogen peroxide also, which makes the madhu antibacterial. Now, the importance of translational research is much the need of the hour, because many of the researchers, many of the preclinical works remain untouched, and they will not be subjected for clinical trials further until and unless, because clinical trials are the gold standards. So until and unless clinical trials properly conducted, the clinical applications and the people or a public, general public, cannot be benefited. So translational research is usually regarded as bench to bedside by ensuring that the scientific discoveries lead to the benefits for the patients and communities. Now, let me start with the work or the research works conducted by me and my team of postgraduate scholars, one after another. The first work, what we did, was the anti-hyperlipidemic and anti-obesity activity of Madhu. That was the experimental activity. Here, we evaluated the anti-obesity and lipid lowering effects of honey processed with triphala. See here, just the shloka mentioned earlier, sthavletu triphala rase. That was the key concept for us to undertake this particular study. And the two components of the madhu, that is kevala madhu, just triphala kashaya. The triphala kashaya mixed with madhu, and triphala kashaya samskarita madhu. The samskara process is already been explained. So these four different things, samples were tried on experimental animals for the duration of 48 days. One more thing I want to specify here: there is no time limit mentioned for the usage of madhu, even for the therapeutic as well as even for the madhu to be consumed as food. What we lack information in our classics is even the quantity of the madhu to be taken as a for diet as well as therapeutic purpose. So maximum duration what we fixed during this study was 48 days. That is one mandala kala generally regarded. And what we observed during this uh, particular experimental work was the weight loss. The weight loss, the treated animals, that is Triphalakashaya Samskarita Madhu, as the book Kaireva Nighantu says, that is Thauletu Trifalarasam, the weight loss among the most significant reduction in the body weight compared to all other groups. That is the Triphalakashaya Samskarita Madhu showed a very good result, not just weight loss, further reduced LDL, VLDL, total cholesterol, as well as triglycerides, because we induced obesity in those experimental animals by feeding them with the high fat diet. So when we tested the lipid profile in those high fat diet animals, we observed a high levels of LDL, VLDL, total cholesterol, triglyceride, etc., etc. And after starting the medicine, Madhu, all groups, yeah, especially in the Triphalakashaya Samskarita Madhu, that particular group, LDL, VLDL, total cholesterol, and triglyceride was significantly reduced. And we concluded that study is a honey processed with Triphalakashaya has a superior anti-obesity and anti-hyperlipidemic activity, not just lowering LDL, VLDL, it increased HDL content. That is more significant. And even the weight of the liver was also reduced compared to the control group animals. That means when we translate this particular information into clinical science, the Triphala Kashaya Samskarita Madhu can be used in uh, the fatty liver, grade 1, grade 2 fatty liver conditions. So this is the chart which describes the reduction in the body weight of experimental animals. The next one, okay, during I missed one thing. This during this particular study itself, what we noticed that the Madhu behaves differently with or without heating. So the two samples were used. That is, one is samskara. So Madhu without heating, that's just mixed with Triphala Kashaya, and the samyoga Madhu without heating mixed with Triphala Kashaya, and samskara Madhu processed with Triphala Kashaya till it turns into avaleha, right? So the Samskarita Madhu was showing this particular activity, anti-obesity and anti-hyperglycemic. But when we compare the blood glucose levels, RBS, random blood sugar levels in the serum glucose levels of the experimental animals, some very interesting factor, what we found that the samyoga, that is Samskarita Madhu, was not having much effect on the blood sugar level, whereas the Madhu mixed with Triphala Kashaya without heating was having the very good impact on blood sugar level in reducing blood sugar levels. So we further continued the study to evaluate anti-hyperglycemic activity of honey in high-fat diet induced diabetes. Here, the experimental animals were fed with high-fat diet, and concurrently the streptozotocin what used to induce diabetes as well. So 8 groups of animals here they were tested, that is high fat induced and as well as streptozotocin induced diabetes, and we tested this particular anti-hyperglycemic activity, because earlier study was anti-hyperlipidemic activity. So here anti-hyperglycemic activity was evaluated for the duration of 30 days of treatment, because earlier study had showed us that 21 days of administration of the drug itself is sufficient to lower the blood sugar levels. Here we conducted the study for the period of 30 days, the total 10-week study, whereas 6 weeks 6 weeks were needed for a successful induction of diabetes in the experimental animals, and remaining days the drug was given, or the experimental drug was given, or a test drug was given to the experimental animals. And what we found was the group treated with honey mixed with Triphala Kashaya, that is Samyoga, showed significant reduction in serum glucose levels compared to the standard drug, as well as comparable to the standard drug, comparable to the standard drug. And the honey processed with Triphala Kashaya did not show as significant reduction in the blood sugar level. That means honey behaves differently when it is heated. So the translational research, what is the conclusion of the research is, for diabetic control, unprocessed honey mixed with Triphala Kashaya is more effective. And for obesity control, the processed honey with Triphala Kashaya is more effective. And one more component we experienced or we observed during this study was the oxidative stress on vital organ was very, very minimal when in that particular group, which is treated with the honey mixed with Triphala Kashaya and honey processed with Triphala Kashaya. This particular effect of organ protective activity we did not observe. So this particular graph is gives you the insight or the observation, what we found, what we noticed, that the blue line or here it is showing that is the standard drug, and the brown line which is showing this particular honey mixed with Triphala Kashaya. That means at par with the standard drug, the effect of the unprocessed honey with Triphala Kashaya, which induces anti-hyperglycemic activity. So honey with Triphala Kashaya unprocessed can be a very good or a better option to deal with diabetic people. And that particular work is still going on in our department. And we are using the honey, unprocessed honey mixed with Triphala Kashaya for pre-diabetes. So likewise, these first study reports suggested that the anti-obesity and anti-hyperlipidemic activity of processed honey, we conducted it to clinical trial also, because the translational research is the need of the hour. In clinical research, we conducted with three different groups of 30 subjects in each group, and we deliberately kept one control group without any medicine, only the food and lifestyle management. This study was conducted for the period of 48 days. Here you can see, among the all three groups treated, again honey processed with Triphala Kashaya is showing very good anti-obesity and weight control, as well as its efficacy is more in reduction in BMI also. So honey mixed with Triphala Kashaya did not show much reduction in BMI as well as the weight. So again, clinically also, this particular concept that the Madhupaka with Triphala Kashaya was established. And one very important thing what we noticed, because I earlier told you have to give some attention, pay some attention to one of the word called as HMF, that is hydroxymethyl perfurol, because hydroxymethyl perfurol is said to be a marker for determining the quality of the honey. So more the HMF means more will be the chances that the honey is heated. So less the HMF refers to, less will be the chances for honey heated. But usually what happens sometimes, when we trust one of such test parameters like HMF, that may mislead us, because for assessing HMF there are two different techniques. One is HPLC technique, and second one is uh, the spectrophotometric technique. Whereas HPLC technique always gives very high HMF content, whereas the same sample when which was tested with the photometric, which was giving the less concentration of HMF. So ultimately, we concluded that the Winkler method, that is photometric method, is more reliable, because the Triphala Kashaya as well as honey, it contains more phenolic content. So these phenolics will interfere with this HPLC type of test parameters by giving more HMF values. Then we moved further. We did, because we observed two different behavior of the Madhu, that is samyoga as well as samskara. With samyoga with Triphala Kashaya, just samyoga with Triphala Kashaya, please don't get confused, because here samyoga means you take Purana Madhu, mix with equal amounts of Triphala Kashaya and administer. Samskara refers to, you take Purana Madhu, take equal amount of Triphala Kashaya, you mix together, heat it till the Paka Lakshanas are observed. This is the basic difference between samyoga and samskara. So the Madhu was behaving differently. When it is unheated, better anti-hyperglycemic activity. When it is heated, better anti-hyperlipidemic activity. And both were not going concurrently. Means samyoga was not anti-hyperlipidemic, samskara was not anti-hyperglycemic. So this behavioral pattern difference, we try to assess what may be the reason, uh, what brings the difference in the components of the Madhu. So we took some samples like aged honey, water processed honey, Triphala Kashaya processed honey, and Triphala Kashaya mixed honey, that is samyoga. The parameters what we took for consideration was HMF, that is hydroxymethyl perfurol, total phenolic content, antioxidant activity, sugar profile, and electrophoretic studies. So what we concluded with this study was the processing honey increases total phenolic content. One thing, very important thing, the phenolic content will be increased. Here, we must know that these phenolics or polyphenols are generally regarded as having antioxidant potential, anti-inflammatory, as well as mild antimicrobial activity also. So, processing, that is heating honey, will lead to increased phenolic content. Fructose also is going to increase after heating process, and the mixing means samyoga appears better for immediate antioxidant effects, because antioxidant activity is also going to be improved, but better antioxidant activity is observed without the heating process. So, uh, next one is very important. After doing all these research activities, we evaluated the polyphenol bioavailability of honey with respect to samyoga and samskara. In this particular study, we evaluated whether the heating, that is madhupak, take by the intestine, intestinal mucosa. This was the objective of the study. Here, the methodology was the ex vivo rat intestine absorption study. Here, the intestinal segments were taken out from the experimental animal, Viscer albino rat, and those separated intestinal segments were used. This is nothing but ex vivo study. The samples taken here was Triphala Kashaya, the Samyoga Madhu in the form of Triphala Kashaya mixed with Triphala Kashaya, that is Samyoga, and the processed variety of Madhu is also used for the study. What we noticed here is very, very interesting. The absorption rate, see, after samskara, after heating, the 67.8% absorption of phenolic compounds takes place uh, within the intestine. Whereas without heating, only 6.8% absorption of phenolic compound takes place. When the water processed honey means the as it is told in Kayadevani Ghantu that for all therapeutic purposes, the honey should be processed with water. If you don't want to process with any other liquid, you can process it with only water. Even after processing it with water, the 24.9% increase in phenolic compounds were noticed. So we concluded the study, that is processing honey with the phenolic rich compounds significantly enhance the bioavailability of the intestinal absorption of polyphenols compared to simple mixing. Heat treatment releases bound phenolics and aids absorption. So, how to translate this particular observation of the research into clinical practice, that if we want to get more benefit of the phenolic compounds, especially the heating of the honey is necessary. Without heating, the honey will not enhance the absorption of phenolic compounds. Now, the next research was to analyze synergistic activity of honey on anti-carcinogenic potential of Triphala. So, it is mentioned, it is established, and Triphala gained more prominence, more importance as the study reports. It was published as Triphala is anti-carcinogenic. So, our aim was to see whether the Madhu in the form of again samyoga and samskara, whether it is having any role in increasing the anti-carcinogenic potential of triphala. So the objective was to evaluate if honey enhances, that is yoga vahitva. Here we use the word enhancing of the karma, that is yoga vahitva, the anti-cancer potential of triphala when mixed, that is samyoga, or when processed, it's samskara. Here the cell lines, it was the cell line study. The cell lines used were cell lines. So what we observed is anti-proliferation. Triphala samskarita madhu showed significant high anti-proliferative activity. That means here samskara of the madhu with Triphala kashaya leads to more anti-proliferative activity compared to just samyoga. So this is in lower concentration, aiding apoptosis. So so it enhances its anti-cancer potential, through validating the Ayurvedic concept of samskara. But here I want to make it, make one very interesting findings during this study. See, the anti-proliferative activity was noticed of as we increase the concentration, the effect that is what happened was, when the further concentration was increased for to analyze the anti-proliferative activity, uh, suddenly there was a drop in anti-proliferative activity. When the concentration was increased too much, there is a drop in anti-proliferative activity. That means the honey or the phenolics may be having too much phenolics are used, or too much phenolic in high concentrations, they may cause antioxidants. The phenolics turn into pro-oxidants. That was established with
Sir,
the next
sorry, sir. ಕೇಳ್ತಿಲ್ಲ
studies.
sir ಸರಿಯಾಗಿ.
So this
ವಾಯ್ಸ್ ಬ್ರೇಕ್ ಇದೆ
we halted. That is honey with triphala. That particular has been mentioned. Shodhan.
ಸರ್ ವಾಯ್ಸ್ ಬ್ರೇಕ್ ಆಗ್ತಾ ಇದೆ ಸರ್.
ಕೇಳ್ತಾ ಇಲ್ಲ.
ಅವ್ರಿಗೆ ಫೋನ್ ಅಲ್ಲ, ಅವ್ರಿಗೆ ಫೋನ್ ಮಾಡಿ ಒಂದ್ಸಲ ಸತೀಶ್.
ಕೇಳಿಸ್ತಾ ಇದೆಯಾ? ಹಲೋ,
ಈಗ ಬಂದಿದೆ ಸರ್.
ಕೇಳಿಸ್ತಾ ಇದೆಯಾ?
ಇವಾಗ ಕೇಳ್ತಾ ಇದೆ.
ಕೇಳಿಸ್ತಾ ಇದೆಯಾ?
ಮತ್ತೆ ಬ್ರೇಕ್ ಆಗಿತ್ತು ಸರ್.
ಇವಾಗ ಕೇಳ್ತಾ ಇದೆ, ಆವಾಗ ಮಧ್ಯ ಮಧ್ಯ ಬ್ರೇಕ್. ಕೇಳಿಸ್ತಾ ಇಲ್ಲ ಮತ್ತೆ. ಕೇಳಿಸ್ತಿಲ್ಲ.
ಈಗ ಕೇಳಿಸ್ತಾ ಇದೆಯಾ?
ಈಗ ಬಂದಿದೆ ಸರ್.
ಎಸ್ ಸರ್, ಕ್ಲಿಯರ್ ಇದೆ.
ಒಂದೇ ನಿಮಿಷ, ಸಮ್ ಟೆಕ್ನಿಕಲ್ ಎರರ್ ಸರಿ ಮಾಡ್ತೀನಿ.
ಒಂದ್ ಸ್ಲೈಡ್ ಹಿಂದೆ ಹೋಗಿ ಹೇಳಿ ಸತೀಶ್, ಬೆಟರ್.
ಸರ್, ಇದು ಸ್ಲೈಡ್ ಅನ್ನ ಶೇರ್ ಮಾಡ್ಬೇಕಾ ಸರ್? ನಮ್ಮ ಕಡೆಯಿಂದ ಸ್ಲೈಡ್ ಬರ್ತಿದೆ. ಸರ್ PPT
ಕೇಳಿಸ್ತಾ ಇದೆಯಾ?
ಸರ್ ಕೇಳ್ತಿದೆ. PPT ಏನಾದ್ರು ಶೇರ್ ಮಾಡ್ಬೇಕಾ ಸರ್ ನಮ್ಮ
Hello?
ಕಡೆಯಿಂದ? ಸರ್ ಕೇಳ್ತಿದೆ, ವಿ ಕ್ಯಾನ್ ಹಿಯರ್ ಯು. ಸರ್ ಕೇಳ್ತಿದೆ.
ನೀಲ್ಕಂಠ್ ವಿಡಿಯೋ ಆಫ್ ಮಾಡ್ಕೊಂಡ್ರೆ ಐ ಥಿಂಕ್ ಸರ್ ವಿಲ್ ಬಿ ಮೀನ್ಸ್ ಇಂಟರ್ನೆಟ್ ಇದು ಯೂಸೇಜ್ ಕಮ್ಮಿ ಆಗುತ್ತೆ,
ಈಗ ಕೇಳಿಸ್ತಾ ಇದೆಯಾ?
ಅವಾಗ ಮಾತಾಡ್ಬೋದು.
ಎಸ್ ಸರ್ ಕೇಳ್ತಿದೆ ಸರ್.
ಕೇಳ್ತಿದ್ವಿ.
ನೀಲಕಂಠ ಅದು ಸ್ಲೈಡ್ ಶೇರ್ ಮಾಡ್ತೀರಾ? ಸ್ವಲ್ಪ ಇಲ್ಲಿ ಸಮಸ್ಯೆ
Oh,
ಇದೆ.
he'll do it.
ಶೋರ್ ಸರ್, ಎಸ್.
ಒಳ್ಳೆ ಇದ್ರಲ್ಲಿತ್ತು. Dr. Neelakanta. Yes.
ಸರ್ ಸ್ಲೈಡ್ ನಂಬರ್ ಯಾವುದು ಸರ್?
ಒಂಚೂರು ಕೆಳಗೆ ಬರಬೇಕು.
Before this. Next. ಒಂದು 3-4 ಸ್ಲೈಡ್ ಆಗ್ಬೇಕು.
ಇನ್ನೂ ಆಗ್ಬೇಕು.
ಯಾ, ನೆಕ್ಸ್ಟ್. ಒಂದು ನಿಮಿಷ. ಬ್ಯಾಕ್ ಬನ್ನಿ.
Yeah. From here you can, because it was breaking down here also, Satish.
ಓಕೆ, ವಾಯ್ಸ್ ಕೇಳಿಸ್ತಾ ಇದೆ ಅಲ್ವಾ?
ಈಗ ಕೇಳಿಸ್ತಾ ಇದೆ.
Okay. So this was the analytical evaluation of honey, what we conducted. For example, to see what changes happen and what is the basic difference between the samyoga and samskara. Why samyoga means unprocessed honey is anti-hyperglycemic, and why processed honey is anti-hyperlipidemic. So then we conducted the analytical study. We saw a basic difference. Even sugar components are going to be changed. Phenolic components are going to be increased after processing. This was the key finding in the analytical evaluation of honey when it was conducted. Next one. Yeah, the key observations, as you can see, the phenolic contents in Samskarita Madhu had the highest total phenolic content. The antioxidant activity is also going to be increased in processed honey. The sugar profile, the fructose-glucose ratio is going to be altered. After samskara of Madhu, there will be more fructose compared to glucose. And we know the when the choice is given between glucose and fructose, the body always chooses glucose first. Having more fructose deprives the body from the glucose. So that may be one of the reason where we can get anti-obesity activity after honey processing. Next slide. So to assess whether honey has a role to enhance the polyphenol bioavailability. So this was done as a ex vivo experimentation by using the isolated intestinal loops from the experimental animals. We used the samples. That's a plain Triphala decoction that the honey mixed with Triphala kashaya, that is unprocessed honey processed with Triphala kashaya. So in that, uh, what we noticed is the Triphala kashaya samskarita madhu, that is processed honey. There will be around 68% phenolic uptake was noticed compared to unprocessed. That means honey enhances the phenolic content uptake by the intestinal loops by around 200 to 300% or more than that. That is unprocessed only below 20% absorption of phenolic compound was noticed, whereas processed honey, it is around more 68% increase in phenolic content. So yeah, previous slide. Previous to that, so once we notice that more phenolic uptake is caused by the processed honey, we even subjected the sample for anti-carcinogenic as well as anti-proliferative activity of honey also. Here we noticed that again the processed honey is having better anti-proliferative activity. The cell line study was conducted here. The colorectal cancer cell lines as well as the breast cancer cell lines were used, whereas the uh, processed honey, here sample showed very good anti-proliferative activity. Very interesting factor we noticed during this study is the usually we think that the effect of the drug will be concentration dependent. That means more the concentration, more the effect. But here, what we noticed, as we started increasing the concentration of the test drug, that is processed honey, suddenly there was a drop in anti-proliferative activity. Means earlier, in initially in lower concentration, it is showing very good anti-proliferative activity, but in higher concentration, the drop of anti-proliferative activity, that means there may be the shift in its behavior of the phenolic compounds where the antioxidant activity turns into pro-oxidant activity. That's why it gives us a clue, or it gives us a basic information that phenolics are not always safe to use in higher amounts. So we concluded this study was concluded by processing phenolic or processing Triphala with honey significantly enhances its anti-cancer potential through synergism, that is Yogavahitva, that validates the Ayurvedic concepts of samskara. Next slide. Here we gave a little break to the concept of Triphala Kashaya and Madhu. We shifted to the next concept, that is Shodhane Haridra Rase, that is Haridra Swarasa, processed the Madhu or the honey processed with the Haridra Swarasa, the turmeric juice. Here, the objective was to evaluate antimicrobial activity of Madhu, that is processed with Haridra Rasa or Haridra Swarasa. The fresh Haridra was collected. The juice of fresh Haridra was taken, and that with the Swarasa of fresh Haridra, the equal amount of honey was added to that, subjected for Madhupaka till it turned into Avalehavat, and that is regarded as one of the sample. And the same sample was compared with only honey, that is Kevala Madhu, and then honey just mixed without samskara, honey mixed with turmeric juice. And even here, two types of turmerics are used. One is the fresh turmeric, The second one is a dry turmeric, because market available turmeric is always the dry sample, not the fresh one, and we are used to we are used for using that dry sample. So organisms used Streptococcus aureus, E. coli, and then Salmonella typhi. So techniques was macro broth dilution assay was conducted. Next slide. Here we notice something very interesting. Next, next slide. Yes. So the best sample among all was the honey processed with fresh turmeric juice. We used here both the samples, that is the fresh as well as dry. We did not get activity in dry turmeric. We just got the activity with the fresh turmeric, because we subjected for the sample for HPLC analysis also. We did not find curcumin content in dry turmeric. The curcumin was seen, curcumin was observed, curcumin was noticed in fresh turmeric only. That means there is a processing which is done to make the haridra more more appealing. That After collecting the fresh turmeric, what the farmers do, they just boil it in water, they peel off the outer layer and then dry under sunlight. So this process will take out the curcumin from it. That's the reason why the curcumin content was not seen during HPLC analysis. Back please. Next. Yeah. Yes, we concluded this study, that is processing honey. Previous slide. Yes, we concluded this study by processing honey with fresh turmeric juice enhances the antimicrobial activity and bioavailability of the curcumin. This validates the concept of, again, Madhu samskara. That means the fresh haridra for the better results of antimicrobial activity. Here, one more thing, antimicrobial activity, what we got, that is minimum inhibitory concentration was 100 micrograms per ml concentration. But what is generally believed, that anything which is having MIC, that is minimum inhibitory concentration, should be less than 50 microgram. Then only the quality of the drug will be much more efficient. But still here, because since it is a herbal oriented, not a synthetic product, the minimum inhibitory concentration, even 100 microgram, can be considered. Next slide. That means here the translational research, the moral of this particular research, to convert it into clinical practice, that honey processed with Haridra Swarasa, fresh Haridra Swarasa, can be used to apply over the wounds, which can prevent the development of bacteria, or it minimizes the chances of infection, because it is antimicrobial. Honey itself is antimicrobial. Adding haridra to it makes it more potent. Then next study was to assess the chemical changes which happens in honey when processed with Triphala Kashaya, focusing on gallic and tannic acids, because gallic and tannic acids are supposed to be the biomarkers of this particular Triphala. And we analyzed the same samples as I was describing earlier, that is samyoga, processed honey, unprocessed honey with Triphala Kashaya, with the help of LCMS and UV spectrophotometer. Next slide. What we noticed, again, the reduction in tannic acid concentration and increase in gallic acid concentration. This means, see, more the gallic acid concentration, more will be the antioxidant activity. More the gallic acid concentration. See, if we believe that antioxidant activity is rasayana, karma, then more the gallic acid concentration, more will be the antioxidant activity. As we mix Triphala Kashaya with honey, there will be a sudden drop in tannic acid concentration and increase in gallic acid concentration. And one more thing, very interesting thing, there are few new compounds which are going to be generated. There is a number of unknown compounds found in sample using LCMS. During this study, the unknown compounds, around 300 to 400 different unknown compounds were there, which were observed, which were newly synthesized during this heating process. Next slide. Now, here the take-home message for the clinical practice. See, we all see that phenolic compounds, phenolic rich, antioxidant, everything. See, Kashaya, we should be a little bit aware of these factors of Kashaya Rasatmaka Dravyas, because Kashaya Rasatmaka Dravyas having Rasayana Karma have these phenolic compounds as a phytoconstituents in them. But there are some adverse drug reactions of these phenolic compounds also. To name few, the phenolic compounds in higher concentration can become pro-oxidants. No doubt they are very good antioxidants, but in higher concentration, leading to oxidative stress, they lead to even cell death, gastric irritation, reduced absorption of minerals, hepatotoxicity with prolonged high dose use, drug herb interactions, hormonal imbalances, allergic and hypersensitivity reactions, cytotoxicity, genotoxic effect of sulfur drugs. These are all will be increased if the overuse of phenolic compounds are seen, or overuse of phenolic compounds, if it is made or if it is done, definitely we can expect such adverse drug reactions. Next slide. Now we shifted our focus to the next compound, that is Gomutra. Is as the shloka told, that is Durmamse Mutra Samyuktam. See, Madhu mixed with Mutra, that is Gomutra, is can be used in Durmamsa. Durmamsa here we considered Durmamsa as a tumor. So honey processed with Gomutra was considered. Again, this was a cell line study. The honey processed with Gomutra was used. Here, the DPPH, that is antioxidant study, was also conducted, and breast and colorectal cancer cell lines were used to ascertain whether the honey processed with Gomutra is having the anti-proliferative activity. Next slide. And definitely, as we expected, same or similar kind of thing, similar kind of result we got. But prominently, again, a very interesting thing here is honey processed with Gomutra has high inhibition on colorectal cancer cell lines compared to the breast cancer cell lines. That is the Srotogamitva of Madhu. Here we can say this is more oriented towards the Annavaha and Purishavaha Srotas. So this unless this is proven clinically, we cannot directly say that it is more effective, but definitely during anti-cancer or so cell line studies, this Madhu or honey has shown very good anti-proliferative activity. So we concluded this study as Gomutra Samskarita Madhu has significant anti-proliferative effect with respect to the cell line studies, particularly on colorectal cancer, which is attributed to increased phenolic content and synergistic activity. This Yogavahitva with Madhu. Next. Right. So this is recently concluded study and submitted to Rajiv Gandhi Health University. See the Kshira and Haridra, as the shloka told earlier from Kayadeva Nikhantu, that is with Kshira, there will be Ropane Kshira Samyukte Shodhane Haridra Rase. So Ropane Kshira Samyukte, for wound healing, the Gokshira Samskarita Madhu, as well as for in Dushta Vrana, that is Shodhana, Vrana Shodhana, Haridra Rasa Samskarita Madhu has been recommended in Kayadeva Nikhantu. So we compared the both activities, whether the samples have the wound healing property, to evaluate wound healing efficacy of Madhu processed with the turmeric juice as well as the cow's milk compared to unprocessed mixtures. Here we tried 4 test drugs, that is processed with processed with Haridra Rasa, Haridra Swarasa, and unprocessed variety of with Haridra Swarasa. processed with gokshira and unprocessed with gokshira. This is the one where the textual reference, as per the textual reference, we did not get the result. Here, the as per the textual reference says the honey processed with gokshira is having good ropana karma, or it must do ropana karma. Here, we did not get the ropana karma, very good wound healing activity with respect to the honey processed with gokshira. Rather, honey mixed with gokshira, that is unprocessed, just samyoga. One more trial, what we did, see, honey, when it was mixed with gokshira, when we tried to apply over the wound induced on experimental animals, because this study was on experimental animals. As you see, the number of groups made were 7: control, standard, zinc oxide as a standard drug, kevala madhu, pure honey mixed, that is turmeric, honey mixed with milk, and honey turmeric processed. So unprocessed and processed differently. Here we had some practical problems also. Honey, when we mix with milk and try to apply, it was very dilute mixture, so it could not be applied over the wound surface. Even after application, sometimes the rats used to lick it up, and again we could not keep more than one rat in cage, because the other rat starts eating the honey which is applied over the its accompanying partner. So the what we did here, we mixed the milk powder, rather using milk gokshira. We tried to mix milk powder with honey and then applied over the wound surface, which gave a better result, or significantly good result compared to the other samples, sorry, other groups. So the conclusion here, what we draw, next slide. So conclusion, that is the milk plus honey, that is only samyoga mixed with honey, is the most effective formulation for wound healing. Processing honey with milk, or the processing honey with turmeric, we do not get that much activity, that much wound healing effect, compared to just mixing honey with milk and applying over the wound surface. So this is again an experimental work, and that will be taken for the next clinical studies as a continuation work. Next, please. So there are few studies under progress. That is honey, effect of processed honey on pre-diabetes with respect to samyoga. This is now going on. Then prebiotic effect of madhu with respect to samyoga and samskara. Here the prebiotic effect means, again it will be analytical study whether the phenolic compounds present in Triphala Kashaya as well as madhu have the prebiotic effect, means they enhance the growth of certain beneficial bacteria isolated from the intestine. So that is prebiotic effect. That study is also going on. The effect of samskarita madhu in sthaulya with respect to route of drug administration. That means the earlier what we used, the samskarita madhu through oral route. Now we want to try it with respect to basti dravya also. So this will be the study to be conducted. And role of Haridas Swarasa Samskarita Madhu in Dushtavrana, the clinical study has to be done. So these studies are planned, as well as the studies are under progress. Next slide, please. So to conclude, so many studies were conducted, and ultimately we can conclude that while experiential or experimental studies provide strong evidence for the therapeutic potential of honey, multiple biological, methodical, as well as practical challenges limit direct replication of these results in clinical research. So translational research is not very easy. So we may get so many positive results with respect to analytical, in vitro, ex vivo, and as well as experimental studies. We get lots of positive results. But when we try to do the clinical research or a clinical trial, the similar effect or similar outcomes we will not see. This is because of so many differences between experimental animals as well as human beings, as well as there will be difficulties in controlling the food habits of human beings also. There will not be absolute control because experimental animals, they will be caged and we are strictly controlling their food habits, whereas in clinical trials controlling human behavior will be difficult. So addressing standardization, dosage optimization and well designed clinical trial is essential for successful translation of the experimental study findings. So with this I conclude my talk. I tried to convey the outcomes, plans of my research are done by me as well as my team of postgraduate scholars in different to ascertain the different types of behavior of this madhu, maybe with Triphala Kashaya, maybe with Haridra Swarasa, maybe with Gomutra in different way. So with this I conclude. I sincerely uh, thank the organizers, the Tribhuvan Holistic, for giving me the opportunity to share some of my experiences in the field of research carried out on the honey. Thank you very much.
Thank you, Dr. Satish Pai, for such a evidence-based and clinically enriching presentation. It was very good. We now proceed to the interactive question and answer session, which will be moderated by Dr. Neelakanta Sajjana.
CK madam, please take over.
Hello.
Yes, madam, audible. Please take over.
Hello.
Yes.
Yeah, okay. Sir, one question is there, how to process honey? I think you have answered it by Dr. Swati. That Triphala Kashaya, samskara according to Kayyadeva Nigantu, I think you have answered it, sir. That was the one question. How to process honey?
In the shloka itself, it has been mentioned, right? Like eight parts of honey and one part of water. See, if no drava is mentioned as such, for all
Yes.
therapeutic purposes, 8 parts of honey, one part of water, you just mix it up, reduce it till the original quantity of honey remains. So this is one way of processing. If the drava is specified, there are 4 drava mentioned in the Kaideva Nigantu. One is Triphala Rasa, they say. Triphala Rasa is nothing but again Triphala Kashaya only. Haridra Rasa, they say, Gomutra, they say, as well as Kshira or Gokshira. There is nothing but. So when the thing comes, these Dravadravas are there, again, the ratio of Dravadravas is not mentioned there. So we took it is Samamatra, Sama Pramana, equal amounts of Triphala Kashaya, equal amount of Madhu. Heating it, or Mandagni till Pakalakshana, we observe.
Thank you, sir. Another question is from Dr. Asha. Triphala Kashaya, honey proportion, duration. I think you have answered it. And Aushadhi Kala.
Aushadhi?
Aushadhi Kala. Actually, I cross-questioned it, what is exactly, but I didn't get the Aushadhi Kala.
Maybe Aushadhi Kala, maybe the whether Aushadha Sevana Kala, maybe the question is about Aushadha Sevana Kala.
Possible, sir.
Yeah. Uh, um, uh, what we did during the clinical trials, we asked the patients or the subjects to take the medicine before food.
Okay.
Half an hour before food, because there is a reason for that. So phenolics usually have a hunger mitigating effect also. So what we need usually in Sthaulya is their hunger to be controlled or regulated. So phenolic compounds have that particular ability. So we advise the subjects here to consume that medicine. Here again, the quantity of medicine, I did not mention how much quantity. See, since there is no reference for the exact quantity of the medicine to be used, the type of the material was avalehavat here. So avaleha pramana was considered. So one pala pramana is the avaleha pramana. So when I try to see what will be the quantity of the processed honey with respect to avaleha pramana, that is 48 grams. One pala refers to almost 48 grams. It came up to 30 ml of processed honey. 30 ml is equivalent to 48 grams. So we gave in divided doses, 15 ml, 15 in the morning times, 15 ml in the evening times, before food. Half an hour before food, it was advised.
Okay, sir. According to Dr. Neelkant has one more question. He is asking which honey is used, the raw honey or the which is available in the market.
No. Again, that is missed. See, the market available samples are always heated samples. They are processed samples because they process, because see, honey, if I just raw honey is kept as it is tend to change its color, it tend to change its odor and everything. So if it is processed, the changes are minimized because the enzymes present in the honey themselves get neutralized when it is heated. It is generally said to be pasteurizing of honey. Say they heat up to 80 degrees Celsius and then cool down. So it neutralizes the enzymes present within the honey. And when the enzymes are not there, the quality of honey remains unchanged. This is the purpose why the honey is heated. But for our study, we collected from BR Hills. The raw honey was used. Again, one more practical problem was there. We contacted for different locations. We collected honey for our studies. When we contacted these honey societies, different honey societies, they were so reluctant to give us the unprocessed honey. They say
Okay.
we process it, we heat it, and then only we send it to market. Then we try to convince it this is just for a trial and practical oriented work, not for selling into the public domain. Then we got the unprocessed honey from honey societies. Otherwise, we don't get unprocessed honey in the market.
Okay. Thank you, sir. There is one more question by Dr. Asha only, that whether samyoga or samskara is better in DM. I think you have already answered. I think it is samyoga, not samskara,
Samyog.
for diabetic
No.
mellitus. Right. Okay. There is one more question, sir. Is there any noticeable chemical change observed in new or 1-year-old honey?
Yes, definitely. In the new honey, there is more glucose content compared to fructose content. In new honey, there is less phenolic content compared to the older ones. It is well established. It is in the public domain itself. As the age advances, means aging process will increase. There is something called as Maillard's reaction. Maillard's reaction is the enzymatic reaction happening within the honey, which increases the phenolic content as the age advances. We have subject, even the HMF content is also going to be increased. See, surprisingly, what we noticed, we subjected 4 different varieties of honey for HMF analysis with the HPLC method. So 1-year-old, new 1-year-old, 2-year-old, 3-year-old, and 4-year-old. In 4-year-old, what we noticed with HPLC method is around 740 milligrams of HMF content per kilo of honey. That is very, very high content. So as the aging process also increases HMF. Again, one more thing. We need not be much worried about HMF.